Hello students, welcome back to my channel AL Bio Tutor. This is Dr. Sumedh. Today tutorial I will discuss about genes and gene mutation. So what is gene? Gene is a DNA sequence which is in a specific site of the chromosome. So you can call it a fundamental physical and functional unit of the heredity. The idea of the gene first was taken by Gregory Mendel. The law of inheritance, which was taken at 1860, Gregory Mendel give an idea about the hereditary factors. Later, scientists found out that these factors they call them G. So, what is locus? Locus is a site of the gene in a chromosome. So, if you take a chromosome, the area, the location, the site of the chromosome which a gene is located, you call it a locus. The alternative genes which is in the same locus, you call them alleles. So we have learned them in these things in genetic lessons, but we have to memorize these things. So this is a chromosome. In a chromosome, the specific DNA sequence, you call it a gene. So if you take a DNA strand as the first part of the figure, it's a gene. If you take the purple color parts are gene and the yellow color areas are which are not genes, we call them intergenic area. Intergenic region as in the green color area, yellow color area. Purple color things are genes. You call them genes. So if you take that genes, so in a gene, there are areas which is not coming to mRNA. You call them, these are not transcribed to mRNA. These are called intron. These green color areas are intron. And the red colors area which has come into the mRNA, you call them as exon. These are the real gene, real sequence of DNA which is given you the specific characteristics. So this is the table of codon. From this codon, AUG, that is the start codon, and UAA, UAG, and UGA, those are the stop codon. These four should be remembered. So every protein starting with methionine, protein synthesis start with methionine, that is AUG, and UAA, UAG, and UGA codon considered as stop codon. So if you take these 64 codons, when you take these 64 codons, there are codons, 4-5 codons which is given the same amino acid because we have only 20 amino acids. Right. So let's go to talk about mutation. So what is mutation? It is the alteration of nucleotide sequence. So if you take the genome is the collectively all the gene of organism. If you take all the gene of the organism, you call it a genome. So is there any alteration of this nucleotide sequence in a genome? Alteration. Different DNA sequence in cells. Alteration of nucleotide sequence is called mutation. Because of the mutation, there's change of the, otherwise you call it alteration in the DNA. This alteration of the DNA will result in phenotypic variation. Sometimes because of the mutations, the color of the flower is white, but because of the gene mutation, it can become red. So mutation, it is the alteration of nucleotide sequence. If you consider the effect of mutation, these effect can be neutral, no any change of the feature, no change of the character, and sometimes there may be beneficial effect, good effect, or otherwise there may be deleterious effect, bad effect to the organism. Right. The way of producing mutation, there are two ways. First one, it is during DNA replication. We call them as spontaneous mutation. We know that when DNA replication, sometimes we can have DNA mismatch. These mismatch will result in mutation. Normally, because of the proofreading activity of the polymerase, as we learned in our previous uh, tutorials, you can uh, watch my videos about the replication, and that video I have talked about it. So with the DNA replication, if there's any mutation, you call it spontaneous mutation. The other way of mutation is due to the mutagens. So if you take mutagens, you can divide them into chemical or physical. Chemical, chemical substance. Some of the chemical substance can be considered as mutagens, or the X-rays, ultraviolet rays. Those are the physical mutagens. Because of the mutations, sometimes there can be carcinoma production. So if because of the mutation, if you have carcinoma, you call them carcinogenesis. Carcinoma is a non-controllable cell division. Cell division and cell growth happening, but this cell division and cell growth, you, that control will be beyond our level. So it becomes carcinoma. When we consider mutation, mutation can be divided into two types again. It can be mutation in the chromosome. It's a big level. The DNA sequence will be changed, alteration of the DNA sequence, and the other one is the gene mutation. In next part of my tutorial, I will talk about gene mutation. Okay, students. So, gene mutation. When you talk about gene mutation, if we think about a single nucleotide pair, you can call it a point. Only one point, so it is a point mutation. So, the mutation can be due to single nucleotide pair substitution. For one nucleotide, another nucleotide will be substituted. You call it a substitution. This substitution will result in three things. With the substitution, there may be a silent mutation, or otherwise missense mutation, or nonsense mutation. Silent mutation. In a silent mutation, the amino acid of production, the resultant mutated amino acid won't be changed. That means, although you have the mutation, the last resultant amino acid won't be changed. So there's no change of the amino acid. There's no change of the protein. So missense mutation, missense means different nucleotide, because of the different nucleotide sequence, there may be difference of the alteration of the amino acid. With this alteration, there may be changing of the protein. So you call it missense mutation. And nonsense mutation, in nonsense mutation, the stop codon will result as the mutation. So because of that thing, premature premature protein will be produced. So one of stop codon will be mutated as a mutation. So because of the substitution with stop codon, protein will be stopped at that moment. So you will result in an immature protein. There are two other ways of point mutation. These are nucleotide pair insertion and nucleotide pair deletion. If we insert a one pair, there's a shifting of the frame. So you will have different amino acid sequence because of the frame shift. Same thing happen if you have a deletion of one pair. We can see it like this. So with the substitution, first you can see the normal DNA and the mRNA and amino acid sequence, and with the substitution, instead of G you have A in the second the substitution. Rather than glutamic, you will result in a lysine amino acid, and results in different amino acid. If you take here in the second figure, it is the insertion of C. New cytosine is coming and results in. So glutamic become glycine, then next glutamic become arginine. So your frame will be shift. There's a difference. And the last one, the deletion is there. So instead of that, your A has gone, the adenine has gone, the fourth adenine has gone. So rather than tyrosine, the result is in threonine. The amino acid sequence will be changed. So there's a frame shift. We call it a frame shift mutation. So there are two examples. We can talk about two examples in gene mutation. First one is the color blindness, that we call red-green color blindness. In this situation, they can't differentiate red and green color. So it's a sex-linked mutation. So that means the gene for the red and blue, red and green. So there are three types of photopsin: red, green, and blue. Red and green photopsins are located in X chromosome, and but the blue photopsin is not in the X chromosome. So red and green photopsins are in the X chromosome. If you have the mutation in the X chromosome, you will result in color blindness. So this is more common in male. Okay. So the next one is the sickle cell anemia. Sickle cell anemia is due to mutation in the gene coding of beta globulin subunit of hemoglobin. As you know, hemoglobin consists of 2 alpha unit and 2 beta unit of hemoglobin. 2 alpha and 2 beta globulin. The mutation is substituted with glutamic acid substituted with valine. Glutamic acid substituted to valine. This will result in alteration of the shape of the protein. So normal beta globulin is the red blood cell with normal beta globulin is, we know that a red blood cells is biconcave shape. But the alteration of the beta globulin with the mutation, you will result in a sickle cells. The red blood cells will have a sickle shape. Because of this sickling, there may be blockage of the blood vessels, and normally we know that red blood cells, the lifespan of the red blood cells is 120 days, but because of the mutated one, they will destroyed at premature time. So there's a reduction of red blood cells and results in anemia. But there's a good side of the sickle cell in a patient. So if someone has sickle cell, they are protected from the malaria parasite. So in a sickle shaped cell, malaria parasite can't live. So they are protected from the malaria parasite. Today We have discussed about gene and the gene mutation. Thank you.